One of Celentyx's USPs is about understanding the human immune system, and not just understanding it, but working with it. Now, how does one tackle human immune system studies pre-clinically? Well, the way to do it is the Celentyx solution. It's to isolate the human immune system from often blood, peripheral blood, but it can be other tissue sources as appropriate, and then to work within the constituent cells of the immune system within those samples and ask how perhaps individually those cells are behaving and responding to a compound, but also ask how those cells are working in the whole mix of the complex immune system that one has. We have created a platform which we call Immuno-Profiling, and it's a platform that's set at different levels. And within each of those, we can create and tailor-make a package that's appropriate to the questions and the issues around a client's compound. So level 1 would be looking at the human immune system in its generality usually. So asking about how a compound might impact the various subtypes in a relatively global way. Asking about proliferation, asking about the differentiation, and perhaps the movement of the different subtypes in terms of their numbers and their survival. Level 2, we start to drill down to more detail. We might, in level 2, isolate a very specific subset, which could be there at very small amounts, yet playing a vital role in the immune system and perhaps the pathologies that derive from it. Now, level 3 would be about, again, examples, because the details will depend upon the client's question, but for example, precise mechanisms of action. So how is a compound affecting the immune system behavior, appropriately or inappropriately? And perhaps even asking about the target. What is the molecular target? Where, within what cell type, is a particular compound acting and behaving? So within Celentyx, we work with normal healthy material and we work with patient's material to fully understand how a compound can act in both scenarios. So depending upon the compound and the pathologies its impact, we can isolate the appropriate cells from the normal healthy individual, but we can go direct to the pathologies that the compound could well be acting on, and we may even be known to be acting upon. Importantly, in both scenarios, both the normal and the patient scenario, we have full ethical approval in place to work with that material from consented individuals. Not only do we have this strategic relationship with our ethical committees, but we are able to mine the data and the information, the clinical information around the patients and the individuals to give back to the customer so that a more complete, rounded picture can be offered and presented as required. So Celentyx has a long-standing strategic working relationship with the Queen Elizabeth Hospital. In fact, the new super hospital based here in Birmingham, UK. The Queen Elizabeth Hospital has access to the whole West Midlands region. That includes a population base which is bigger than any other health authority within the UK and possibly even within Europe. This population base is highly diverse. In terms of, for example, ethnicity, we have a broad representation of virtually all the ethnic communities within the West Midlands region, and they are coming for their specialist treatment for their immune pathologies to the Queen Elizabeth Hospital. And that working relationship is not only in terms of access to the patient samples, which of course is vital in the work that we do within Celentyx, but also occasionally for the interpretation of the data. So within Celentyx we have the medical expertise around the immune pathologies that we are interested in and our partners are interested in. Within Celentyx, we have state-of-the-art technologies, state-of-the-art equipment, and state-of-the-art ability to analyse those very unique subsets and how they interact with one another. That isn't where the Celentyx solution stops, far from it. In fact, at the very beginning, we work very closely with our clients to address the questions and issues that they have before we start to design the experiments themselves. This can sometimes be through receiving the data package that's already been generated on a compound. And we can look at that in the round and within the immune cell context, and then suggest the appropriate ways forward as to how to better understand what the compound might be doing within the human immune system. And then as data starts to be generated from ourselves, we will then feed that back to the client, and in an iterative process we will then design the next best step, the way forward from there. And so it goes. And then when we have the full package of data at the end, it doesn't stop there either. In fact, that's where the expertise is really brought in and comes to the fore. The ability to interpret what we've discovered. And again, from that interpretation, we will suggest the best next way forward, the best next steps to perhaps understand fully what a compound is doing.